Okay. Hi, everyone. It is my great pleasure to introduce you all to our speaker today, who's one of the world's leading experts in the study of fundamental B cell immunology. Dr. Shiv Pillai is a professor of medicine and at the Health Sciences and Technology at Harvard Medical School, and he's also a professor at the Reagan Institute. And he is currently the director of the Harvard Graduate Program of Immunology. Dr. Pillai completed his medical studies in India and subsequently completed his postdoctoral training with David Baltimore at MIT. It was at David Baltimore's lab where Dr. Pillai discovered the existence and importance of surrogate light chains for B cell development. So we are extremely grateful for having you today, Dr. Pillai. Thank you so much for joining us, and I look forward to your talk.
Thank you, Kiwi, and it's good to see a lot of familiar faces. This is my third talk of today. So some people have heard one version of the more detailed version at 12:30, which we published quite recently, but I'm actually going to talk about a few other things as well. And maybe I could have put back one or two more slides, which I can get to if you let me just share my screen and then I'll. Okay, so I'm going to actually say some things on the side, which may be a little more scientifically complex, but in general, this talk is going to be pitched fairly low to begin with. I'm going to give you a broad overview of immunity and vaccines to begin with. I'm going to talk about the pathogenesis of COVID-19, and here we can go in any depth you want. We can talk about things where they stand. And I will talk a little bit about our current research as well. And I will end with something called Quietly into the Night, the COVID version. Okay, so most of you are aware that SARS-CoV-2 binds to ACE2. There's a protease that's involved in cleaving the spike protein of SARS-CoV-2, and then that's how it enters the cell. And once the virus enters, if it gets into the lung, it might infect type 2 pneumocytes, and you have an infection that's set up. If when you talk about immunity, now there's a lot of people, for you this is not going to be new information, but we do think of innate and adaptive immunity. And adaptive immunity we break up into humoral, which is antibodies, and into cellular immunity, which is made by T cells. I think COVID-19 is largely a disease of innate immunity. And I don't think there is that much of a role for adaptive immunity in the actual disease process. Of course, there are events, and I'm going to talk about events, and we've described them, but I think the real driving forces are innate, and we can talk about that. One of the features of innate immunity that we will talk a little bit about are type 1 interferons, and I might get into a little more depth about it, though I haven't included all the slides on that. And so we have barriers, we have cells that are relevant, and then we have cytokines. These are all part of innate immunity that we just want to touch on. Okay? And innate immunity happens soon, adaptive immunity happens late. So antibodies protect us in many ways, but the way that is most relevant in the context of vaccination, I'm going to start with just the principles of vaccination, is that antibodies neutralize. And so neutralization here means generally for vaccination, if either the antibody binds to a toxin and prevents it from binding to its receptor, or the antibody binds to a virus and prevents the virus from entering a cell because it cannot bind to its receptor, that is what we call neutralization. And there are other functions of antibodies, which I won't get into right now. And just to remind you that we have about 500 billion 500 billion lymphocytes. We have a very large number of lymphocytes. About half of them are T cells and half of them are B cells. But at the end of the day, we need to develop memory. So the goal of vaccination is to choose a few of these guys who recognize what we want them to see. So in this case, it might be the receptor binding domain of the spike protein, and we want to create memory. So the major goal of vaccination even today is to make long-lived antibodies that will prevent us from infection. We've thought maybe we should just make memory cells. Memory cells won't protect us from infection alone. we need to make long-lived antibodies, and the immune system is designed to do both these things. And the key to making long-lived antibodies to actually activate both T cells and B cells. So while I say that, adaptive immunity doesn't have a big role to play in severe disease and the pathogenesis of the disease, if we are able to be vaccinated, it'll be adaptive immunity which will give us vaccine efficacy and protect us in the future. And one of the reasons why this infection has been so terrible is that as a species, we didn't even have any cross-reactive antibodies to protect us in the first place. So there are a number of sentinel cells in the immune system. So mast cells and macrophages can set up inflammation, and dendritic cells are the ones that take the information from a tissue site. So this could be a vaccination site, and take it to the nearest lymph node. And it is in lymph nodes that we're going to initiate immune responses. So in the lymph node, just providing something foreign on an MHC molecule is not enough to activate a T cell. We need also to have something saying that you have come from a land of danger, that there was something pathogenic around. And so this molecule here, the costimulatory ligand, is required to provide a second signal to activate a T cell. That means, again, in vaccination, you need to have a proper adjuvant, which is going to activate the dendritic cell to make the costimulatory ligand, so that at the site of vaccination, the immune system must think, there's something dangerous out here, I need to make a response. And that is where most of our vaccines aren't terrific. We are great at adjuvantation when we completely borrow something from a virus. When we try to do it on our own, we are not as great. And we can talk about this in some depth if you want, okay? So we have signal 1, signal 2 need to be generated. And at the end of the day, you want to make antibodies, and antibodies are going to bind to, they're going to bind to an antigen. But we do need to have, during an immune response, just as pathogens go through mutation, the only response we can use to make better and better antibodies, which will bind more tightly, and to make memory. In order to make really long-lived antibodies and to make memory, we need to go through a Darwinian process. And the Darwinian process is something we call a germinal center. So we start in an immune response, and I'll show you our data showing that in COVID-19, this process can be destroyed, okay? So in a normal germinal center, we mutate B cells. So if we had a B cell that recognizes an antigen, maybe not too well, they mutate. A clone might mutate into multiple cells. Each cell acquires mutations which might make the antibody better or worse at binding to the original antigen. Then we test these cells in the light zone of the germinal center. If the cell binds well, we actually send it back to mutate more. And this goes on a few times. And eventually, at some point, when the cell has been mutated multiple times and has become better and better, and maybe has gone from 10 to the minus 6 to 10 to the minus 10 affinity or 10 to the minus 12 affinity, at that point, the cells earlier on get released to become memory, and later on, when they're mutated even more, become plasma cells. Now, these plasma cells, if they home to the bone marrow and come from the germinal center, we believe they will be there for a very long time. With a yellow fever vaccine, they will be there for 120 years. That's their half-life. Okay, they're really forever, for your lifetime. With the flu vaccine, as I just told some people who were in class with me this morning, it's only for a few months. You can give someone flu vaccination and you don't get a response to last for more than a few months. There's a paper that just came out from Rafi Ahmed's lab, which showed that with the flu vaccine, you do make plasma cells that go to the bone marrow. But if you look after some months, there are no more plasma cells that are making anti-flu antibodies in the bone marrow. So they're not long-lived. They probably never went through the germinal center. There's not an increase in somatic hypermutation, the process by which you get higher and higher affinity antibodies. So the flu vaccine itself, to our mind, is working somewhat like COVID-19. It is not able to make this response. And so for multiple viruses, including COVID-19, Unlike measles and mumps, where you get memory for life, antibodies for life, SARS-CoV-2 is not that kind of a virus. It's behaving much more like our standard flu vaccine. So at the end of the day, you want these high affinity antibodies that can bind tightly to your antigen. And we do get pretty good affinity antibodies in COVID-19, but we don't have durable antibodies. And the data is coming in. Even today, there's more data coming in. So up to 5 months, there are 2 or 3 studies now showing that the antibody levels keep declining. And this was known in SARS and MERS, that for the majority of patients, antibody levels decline. In a small percentage, they continue for longer, and I think we know why that happens. Okay, so when you think about vaccines, what must they do? They must induce signal 2 and give you good adjuvantation. They must create a germinal center response. They must have somatic hypermutation. And the whole point is to make durable neutralizing antibodies. A number of protein vaccines that we use today do not do so. Toxoid vaccines are better. Tetanus toxoid, diphtheria toxoid, they last for 5 to 10 years of protection. With live attenuated viral vaccines, which have other issues with them if you're immunodeficient and so on, then you get really long-lived immunity. So we still don't understand everything. So conventional vaccines, the approaches are either you took the live virus and then you heated it with chemicals, and there are some viruses, some vaccines now for COVID-19 that have been made this way. So it's just a chemically or heat inactivated vaccine. This is a killed virus. This is the vaccine we use for hepatitis A. This is the vaccine we use for flu and polio and for rabies, this type of vaccine. There's another type of vaccine where you have a toxin and you make it, you actually denature it to make a toxoid, and then you adjuvant it a little bit, and then it becomes decent. This is what we see with diphtheria and tetanus. And then we have subunit vaccines, which are like hepatitis B, pertussis, shingles, pneumococcus. These are all protein vaccines. They tend not to be that long-lived. And of course, the live attenuated viruses, these are the ones that give us the best immunity. So MMR will give you lifelong immunity, almost very long immunity. Rotavirus, chickenpox, yellow fever. This category of vaccines works well. They are live attenuated. Of course, there's some theoretical risk of reversion, but that's taken care of by making so many mutations that allow the virus to still replicate so that it can extend the time in which you're getting immunity, but you don't have these viruses actually cause disease. Okay. Now, for COVID-19, there's a couple of new types of vaccines we're looking at, and they're not unique to COVID-19. They came earlier on. So one of them is the hybrid vaccine. And a hybrid vaccine, one example of it is the adenovirus vaccine. And an example that's done at Harvard is with Ad26 by Dan Baruch's group, where basically here you're taking a non-replicating adenovirus, you strip it of some essential genes, and you in that space, you can put in a lot of things into an adenovirus. And in that space, you have put in, let's say, the spike protein of SARS-CoV-2. So this principle was used by the Harvard Janssen vaccine, the Oxford AstraZeneca vaccine, where they used a chimp adenovirus. CanSino Bio used Ad5, but they probably should not have, because we have known for a very long time that Ad5 based vaccines, they're already antibodies to adenovirus 5 in most humans. So about half of the people are not going to even take this vaccine because they have antibodies against it. So there wasn't a very good choice. And not surprisingly, some of the studies with this vaccine have not been as effective. Okay. Now, whether there will be problems in the long haul, hybrid vaccines, though they've been made for the last 7 or 8 or 10 years now, they haven't been effectively used in large populations yet. There's a big trial in Durban from Dan Baruch for HIV, but overall the use is more limited. So whether there will be toxicities that we don't know about, there have been early reports about the AstraZeneca and maybe transverse myelitis. We don't know. We have to wait and see. Okay. Of course, the more interesting vaccines are, so the DNA vaccines haven't yet taken off, but RNA vaccines have. And there are two major RNA vaccines for SARS-CoV-2. One, most of you know, is the Moderna vaccine, and the other one is BioNTech or Pfizer. So BioNTech is the German company that made the vaccine. Pfizer bought off the vaccine and is doing the trials. So in these cases, they're using a messenger RNA. The messenger RNAs are modified. So in order to reduce some of the toxicities, they're just putting a messenger RNA, which has something you don't know in it, like a spike RNA. But beyond that, these are fairly immunogenic, and they're also in a nanoparticle containing capsule and so on. So there's some adjuvantation. Again, we don't know how long-lived immunity will be here. I know from Dan that there is immunity, at least for some of the adenovirus vaccines that has been looked at for about a couple of years. So I'm hopeful that will be a little longer lived. We really don't know with RNA vaccines what will durability be like. Actually, all of them are doing studies which don't address the question of durability. They claim to look at TFH cells in the blood, but that is not going to tell them about durability. I'll tell you why if you have questions. Okay. So obviously, if you get infected and you're a susceptible person, maybe it's something. So already we talk about type 1 interferon, the lack of, either the presence of antibodies to type 1 interferon. There's been a nice study on that. There have been a bunch of individuals, about 10% of people who get very sick have been shown to have antibodies to type 1 interferons. So break intolerance. About 4% of the people who get very sick have been shown to have pre-existing, mutations in this pathway that makes or uses type 1 interferons. So these are two studies from John Laura Casanova. So for about a sixth of patients, we have a good idea about why they're susceptible. Why are you susceptible when you're old? We have many theories about, immune senescence and so on, but no hard evidence beyond what I've told you. Maybe they break tolerance more and they have more antibodies to type 1 interferon, but that still accounts only for about 10%. Another possibility, of course, is that there's a lot of people with diabetes, hypertension, so pre-existing conditions. And there the thinking is that maybe your inflammasomes are more ready to go. The inflammasome is part of innate immunity helps you to, is the process by which we actually secrete IL-1 involves the inflammasome. So it might be that patients who are more susceptible have already got some level of cholesterol crystals or some other trigger activating the inflammasomes in them, and that makes them more prone to have a little stronger inflammatory response in COVID-19. And it can come back to that as well. So inflammation, this is this is not COVID, but in an infected lung, you have a lot of immune cells coming to the lung. In normal heart, this is what it looks like. Even if you don't have an infection, but you have myocardial infarction, you have inflammation. Now, COVID-19 is a disease which would not exist without inflammation and the innate immune system. It is actually the end result of all the damage we see is because of the innate immune response to this virus, which has managed to subvert the type 1 interferon mechanism. Okay? So let me just talk about one or two things. I want to say that one of the things you would see in COVID-19 is this overall an increase in myeloid-like cells, which are cells like neutrophils and macrophages and so on. And there's a decrease in lymphoid-like cells, which I'm including PDCs in for many good reasons. ILCs are down. NK cells, which normally go up in older people, are down in number, dramatically down. So there's a lymphopenia, and lymphoid-like cells are decreased. Myeloid cells are increased. So there's an imbalance that's created. The type 1 interferon system exists to actually protect us from viral infection. And what I'm going to tell you, I'll just mention a couple of things that the virus does. But the virus has many tools by which to prevent our own sensors from seeing the viral RNA and making type 1 interferons. And as a result, this cripples the initial response that we have to the virus. In fact, 1/3 of the known genes of the virus are involved in crippling the type 1 interferon response. So just to give you one or two examples, so in a normal messenger RNA, we add a cap at the 5' end. We call that the 7-methylguanosine cap. We also, as part of the capping process, we add a tassel. And the tassel is a 2' O methyl group that's added to the first base, to the ribose attached to the first base, which is not the cap that's added. Okay, so these two events are both crucial to subvert innate immunity. So just to explain what this is to you, if this is the cap, so this was the original last base of the RNA over here. It's attached to this ribose. This ribose is going to get a 2' O methyl. This is what I'm calling the tassel, okay, on the ribose. And this entire end is going to have added to it a cap, which is the 7-methylguanosine, with a methyl group again on the guanosine itself. Okay, so this whole thing is the cap 0 structure, and this is the cap 1 structure. And I'm going to tell you that this and these two modifications exist primarily to subvert innate immunity when the virus does this. Our RNAs do this, so we can discriminate between our RNAs, our messenger RNAs, and viral RNAs. So viral RNAs, in theory, should neither have the cap, nor should they have this tassel. And then we can say, that's not ours, we should respond. Whereas our RNAs have these two modifications, and we are protected. So the reason is that we do have these guys, RIG-I, which is a sensor in the cytosol, and MDA5, which is another sensor in the cytosol. So RIG-I sees some short viral double-stranded RNAs, and MDA5 sees longer double-stranded RNAs. But RIG-I needs that the 5' end of the cap be a triphosphate, and there be so the 5' end of the RNA be a triphosphate, and there be no cap at all. That is, no cap 0 structure for RIG-I. MDA5 can tolerate a cap, and it can still respond. But the moment you have a tassel, when you add the 2' O methyl group, then MDA5 can no longer bind. And so these two guys are going to be subverted by the virus. And so this is just to tell you that the virus has a few genes. One of the genes is going to be this NSP14, which is going to help add the cap. There's more genes involved. I'm just mentioning one here. And NSP16 is going to add the tassel. And so you just think about it. The virus comes in, you start making viral proteins. The viral proteins then inhibit the cell from responding to the viral RNA, in part by putting on these modifications on the viral RNA, which is then protected. This is an image. It's a very beautiful image of what happens during coronavirus infections to our endoplasmic reticulum. What you see here is your normal endoplasmic reticulum, which buds out to form these big vesicles. And these vesicles, these are double membrane vesicles. They're like balls, double membrane balls, within which the virus is replicating. The virus doesn't want to be seen as double-stranded RNA. So even though it's also modifying, the RNAs so that you want to activate RIG-I and MDA5, it's trying to take no chances. So when it's multiplying, it wants to multiply free. And it makes the cell, again, 2 or 3 viral proteins are involved in this. They make the cell cause the endoplasmic reticulum to bud out, entrap the virus, and this is where they start to replicate. Now, was puzzling for some years. It's all very well, how is the virus is there? How does it come out? And what was shown recently, there's a beautiful cryo-EM image. Again, this is a particular protein of the virus called NSP3, which actually makes a beautiful structure. And this beautiful structure actually serves like a pore. And the presumption is, again, the formal proof is not there, that this pore on the double membrane vesicle allows the viral RNA to come out. And then it's being protected by then, because a lot of the other enzymes are going into this double membrane vesicle, and it's protected by not having the availability to activate either RIG-I or MDA5. Okay? So another protein of the virus called NSP1, it blocks protein translation. It actually, this NSP1 enters the ribosome entry site for messenger RNA so that host messenger RNA cannot enter. Nevertheless, this protein manages to talk to the 5' UTRs, untranslated regions of the viral RNAs, and usher them in. So it's blocking host messenger RNAs from coming in, making type 1 interferon, among other things, but it's allowing viral messenger RNAs to come in and be translated. Okay, so these are just some mechanisms. There are a number of mechanisms by which you're evading, the virus is managing to evade innate immunity. And these are some of the reasons why this virus is extremely virulent and can completely shut off your ability to make type 1 interferons. However, you could have, so if I had someone with a mild infection, what's going on? They are actually being protected. How are they being protected? So if you have a mild infection, okay, and let's say it's a relatively small amount of virus, and there's pretty good data now, it's epidemiological, that a low viral burden at the time of infection ends up with mild infection. When this virus, if this virus got into a particular cell, let's say got into this cell is not going to make type 1 interferon. The virus is going to cripple it from that perspective. This cell is going to shed virus. But early in disease, when all of this is happening, you actually have plenty of plasmacytoid dendritic cells around. These plasmacytoid dendritic cells can make a lot of type 1 interferon. They make 100-fold more type 1 interferon than any other cell. And they can make enough type 1 interferon to bathe the neighboring tissues and squelch infection. You could also activate some CD8 cells at this point and come and delete the infected cells. So in a mild infection, in someone without susceptibility, even though the virus still has the potent power to prevent type 1 interferon induction in a cell it infects, when it gets out, it can be taken up by a PDC, if there are enough PDCs around, and then it can induce an immune response that is going to include the type 1 interferon response, which could be protective to a great deal, and then you could have CTLs come and clear up. Now, this is what we would like to see, where you have the antiviral state is being favored over inflammation. And you're not getting to the severe inflammation because the viral burden's reduced. The amount of viral material, which you call PAMPs, is now not that much. And you can keep things in check. And some people may be asymptomatic. Some people may get mild disease. Okay? So that's one view, and we'll come to what happens in severe disease in a bit. So in severe COVID-19, what we want to think of is inflammation is completely out of control. It's heavily revved up. You might say that to begin with, the macrophages in this person and the neutrophils in this person were more primed already because they have all these metabolic changes because of other pre-existing conditions. But then they get completely out of control when they are seeing a lot of viral products. I'm seeing a lot of what we call DAMPs. When you get cell damage, you create things called DAMPs, and then this can cause even more activation of innate immunity. The antiviral stage is largely evaded. It is evaded even more in severe COVID-19 because the moment you get more inflammation, you are now causing the reduction in the very cells that can protect you, which are PDCs, which could give you good type 1 interferon, and NK cells, which could also cause some death of the infected cells. So there's this balance that gets lost. Okay? So what triggers innate immune mechanisms that are damaging in COVID-19? And what are the triggers? How does this happen? The way we think about it is that, of course, there's viral RNA. And viral RNA is now, instead of doing two things, instead of activating on the one hand type 1 interferons, or the other hand inflammation, it's actually doing only the inflammation. And then the inflammation is going out of control. So this is what tends to happen first. But their viral RNAs can activate both. The N protein might have a role. There is an unpublished report out there, so I'll hesitate to go there. I talked to some students today about what's been published about the spike. So the spike protein, there's at least a nice paper that's out, has been shown to activate the alternative pathway of complement. So we could have complement activation by the spike protein. And then there is a lot of death and damage. So you're going to have DAMPs coming out of unhappy cells. And that might be one of the big drivers in COVID-19, that you have a lot of cell damage, and this propagates the inflammation more and more, especially when you don't have any feedback control. Okay, so I mentioned this before. I just want to point this out, that severe COVID-19 is linked to defects in the type 1 interferon pathway. So we do know that you have, if you have defects in type 1 interferon in terms of mutations affecting these cells, that can contribute to this problem. And this is about 4% of people. And if you make autoantibodies against type 1 interferon, that accounts for about 10% of severely ill people and for 0% of moderately ill people. So this is pretty telling that this is the distinction. And why do viral PAMPs not trigger protective antiviral response? And again, I've sort of mentioned this. The main reason is that you don't have too many PDCs around. So both our group as well as the group of Florent Guinhot showed in very different ways that PDCs are not myeloid cells. They come from progenitors that are more lymphoid-like. So PDCs actually go down. Now, you can ask me the question, why they go down, we can talk about it. PDCs go down in COVID-19, NK cells go down, innate lymphoid cells go down, B cells, T cells, they all go down dramatically. And in the study that we did show that if you looked at the earliest known B cells, which are transitional B cells, they actually dramatically reduced in severe COVID-19. And this tells us that it's probably at the bone marrow level that we're suppressing cells from coming out. There's some activation to cell death of some cells outside, but for the most part, this reduction is either the bone marrow or thymus level, where you get some lymphopenia and very severe T cell depletion. From a bigger picture view of the disease, this disease involves many things, and one of the things that does involve severe disease is coagulation. So you have a hypercoagulable state. You form blood clots in certain blood vessels. This is not the same picture that you see in disseminated intravascular coagulation, which I think at least Omar and I was wondering, maybe there was someone else who should know that as well in the audience, should know that this is not what you see with gram-negative septicemia. But you see coagulation, you see a blood clots forming in certain blood vessels to certain organs, and then you get severe organ damage. So this process, in which you have either you have the virus infecting endothelial cells. In some circumstances, it does that. When you damage an endothelial cell, it releases a protein called tissue factor, which appears on the surface of the cell. This allows platelets to collect and start a clot, initiates the coagulation pathway, and you can get clot formation. So you can also have activation of complement, shown over here, and I'll come back to that. And the activation of complement and inflammatory fragments of complement might also damage the endothelium. They may also activate neutrophils to extrude their DNA and the granule contents and form nets. And this can start to occlude the vessel, but the net proteins can also, the granule proteins can also damage the endothelium. So once you get endothelial damage, you're invariably going to get clotting. So in certain places, you're going to have inflammation, you're going to get clots form, and the organ is going to be more and more compromised as a process, as part of this process. Okay. Now, we also have neutrophil migration and netosis. We also have complement activation and hypercoagulability. And the reasons for this is, of course, I said complement activation. I'm going to explain on the next slide how that actually happens. But complement itself, when you activate the complement system, you can partly activate coagulation. When you activate the coagulation system, you can partly activate complement as well. There's some overlap in protease activities. So this might be part of it. You also have the acute phase response, allowing the liver to make huge amounts of fibrinogen and clotting factors and so on. So all of this together might contribute to the hypercoagulable state. On the basis of one paper that's come out recently, we have a better understanding of how complement might be activated in COVID-19. And this is actually a paper from the Brodsky lab at Johns Hopkins. And basically the argument here is that in normal circumstances, a protein called factor H binds to heparan sulfate on the surface of endothelial cells, for instance. This factor H is able to bind to any C3b. This is the most important component of complement that can be spontaneously created in the blood. So it's C3 that got cleaved to C3b and covalently attached to the cell. So factor H can bind to C3b and set it up to be a target for a protease called factor I, which comes and cleaves C3b and destroys it. Okay? So this is what happens normally. And normally in our blood, we don't activate complement because one of the ways in which we inhibit complement is to have factor H bind to activated C3b and slow it down. Okay? And C3b can be formed spontaneously at some low level. In a COVID patient, the spike protein on the virus itself has a pretty good affinity for heparan sulfate. So now we are displacing all this factor H from the heparan sulfate. As a result, you cannot destroy C3b anymore. And if you're not destroying C3b that efficiently, then C3b can bind to the rest of the proteins relevant to the alternative pathway. In this case, is factor B, which will get cleaved by factor D. And this forms the C3 convertase, which is going to trigger complement activation quite significantly. So we got significant complement activation with SARS-CoV-2, and normally in controlled circumstances, our inhibitors work well for us, like factor I. Now, this is one of the reasons you want to keep in mind that this might be a major trigger in COVID-19. I'm not saying it is the main trigger. We need more studies for people to look at it, but it may be a major trigger in that you activated complement fragments could activate neutrophils, among other things, and then it all builds from there. So the big picture view of the pathogenesis of COVID-19 of severe disease would be that you have increased inflammation, but a decrease in the antiviral state, an increase in myeloid cells, but a decrease in lymphoid-like cells, an increase in pro-inflammatory cytokines like IL-6 and TNF and so on, and a decrease in type 1 interferons. You have an increase in complement activation, in netosis, in coagulation, but you have a decrease in tissue integrity and organ function. So this is how the disease is being set up. Now, specific innate changes in lymph nodes may also impair adaptive immunity, and that's what has come out from some of our work. So let me take you through one or two things, and I apologize to the couple of people in the room who were in class today because I did a more detailed version of this. This is the last bit of my talk. Okay? So what we showed is that in patients with COVID-19, and we compared them with controls who happened to unfortunately pass away at the Brigham at the same time, they were age matched, and we looked at lymph nodes, and you can see these beautiful golden BCL6 positive germinal centers in the controls, and we quantitated all this. And I'm not showing you the quantitation, it's in the paper, but in the disease, these are rare, okay? You can't see BCL6 positive cells at any great density, especially if you look earlier on. If you look earlier on in the control, there's lots of nice germinal centers. Again, we have perfect quantitation because we use some approaches where you can quantitate. Early in disease, we break up our COVID patients to those who unfortunately passed away very soon and those who survived a bit. Early in disease, there's very few BCL6 positive cells. And as they're trying to recover and go through convalescence, but unfortunately they didn't make it, maybe like 1.5 months later, there are some BCL6 positive cells, but when you quantitate them, they're way down. Okay, and I showed the students who came to class this afternoon all the quantitation, but I just want you to keep in mind, we have been able to quantitate all these things and look at a number of patients. So why does this happen? Why do you lose germinal centers? We knew from a number of interesting mouse studies that in severe infections with malaria, with a particular rickettsia, with a model of unusual model which we have recreated in the lab using LCMV with the pre-immunization step. In all of these cases, you lost germinal centers. In the first two cases, they were able to reverse this loss of germinal centers by using an anti-TNF antibody. So we started to wonder, maybe it was because of too much TNF being made locally in the lymph node that perhaps this was leading to this block in differentiation. So what I haven't told you here is that there's a specific block in T follicular helper cell differentiation. There's an increase in cells called Th1 cells. There's more of interferon gamma, and there's a block in T follicular helper cell differentiation. They will not get to the stage where they make BCL6. So why is this happening? We showed there's good TB collaboration. T cells are meeting B cells. T cells are activating B cells outside the follicle. And there we are getting class switching. We're making antibodies. This is where you make your anti-COVID-19 antibodies. All this is happening. We looked in the blood of a number of people, showed they have nice antibodies against the spike. So they can make antibodies. Okay, it's not that they can't make, and they have B cells that can see the spike. The question is, will they live very long, and will you have long-lived immunity? And to see whether this TNF model fits the hypothesis, based on the mouse studies, we do show that there's a lot of TNF in the lymph nodes in disease, just TNF in the middle of the follicle in controls. When you quantitate this, a lot more TNF expressing cells in patients with COVID-19 and far fewer in the controls. Okay, so we think this may be the mechanism. If I had to give you a big picture view of the mechanism we try to put forward in our story, we say that when you have a normal infection or measles, or when you have a normal vaccine that actually works, like the yellow fever vaccine, you probably make good numbers of Th1, Th2, Th17 cells. You have T cells that get activated and move towards the follicle. T cells then activate some B cells, which are selected B cells which have seen antigen. Then you make a extrafollicular focus, some B cells multiply. B cells then contribute to T cell activation, and then you make T follicle helper cells which express BCL6. What we showed in COVID-19 is these cells aren't made. These cells are not there. So normally you end up making germinal centers. In patients with COVID-19, you have an excess of Th1 cells. You have a lot of TNF secreting macrophages, and you actually don't get the state of germinal center formation. They are lost. Okay, so we think pre-existing germinal centers against normal flora are collapsing because of the milieu changing. And though you have huge amounts of viral antigen around, we've been able to have, in these same humans, there's been viral RNA being shown in the lungs at that time. In spite of having a lot of viral antigen, you can make immune responses, but you cannot make probably durable immune responses because you don't make germinal centers. So the big picture view is that early in infection, in someone who's got severe disease, didn't control the virus very well. Type 1 interferon couldn't do it. The viral load might have been high. And all these evasive mechanisms worked against those people, unfortunately. There's activation of the adaptive immune system in the thoracic lymph node. That's what we look at. But there's also, as a result of the severe inflammation, severe lymphopenia. Everything is down in numbers. There's a depletion of CD8 T cells, which we think is actually due to the TNF, because we have gone on now to show, this is not published, we've shown this is all activation-induced cell death, that these cells are being induced to die, CD8s in particular. And there's a collapse of germinal centers. Earlier on, almost all the inflammation is very much just innate cells. Later on, if you look after a few weeks, then you start to see a role for some T cells, though the numbers still are not yet great by then. There's a number of T cells that are activating the macrophages and so on. in the course of trying to eliminate the pathogen from the lung. Okay, so that is the big picture story. I can take questions and then maybe go to quietly into the night, and maybe let me just do that, and then I'll go to quietly into the night.
All right, sounds great. Thank you so much for that amazing talk, Dr. Pillai. So we have a bunch of questions, and okay. I'll start with this one. So I think someone from the audience asked, is there a way to maybe screen for patients with mutations that are unable to make type 1 interferons in order to be able to assess the risk for COVID-19? And what do you think about that?
I mean, in theory, that's of course possible to do. I mean, if you get everybody's genetic information, you would do that. It's kind of expensive to do these kind of things, but in theory, that could be done. So it's a combination often of age and maybe susceptibility factors like this, but
Okay. And I think
are people allowed to ask the questions directly? No.
I think they can. Who let me just see. So I think
Yeah, I mean, you can just
yeah,
say, ask
why
the question, and they will unmute their mic and they'll ask the question.
yeah,
You might even
Dorami.
put their video on for all you know.
Dorami, do you think you want to ask your question?
Can people hear me?
Yeah, a little louder if you can.
Yeah.
Okay, yeah, my mic has been going on and off. So going back to earlier parts of the talk when you talked about the different mechanisms that the virus uses to evade the interferon pathway, which of those are like unique inventions by SARS-CoV-2 and which are common tricks by coronaviruses?
No, most of them are common to the severe coronaviruses. So some of this was known from CoV-1, SARS-CoV, right? But not studied as much because people didn't get so excited at that time, right? So there were already studies that were coming out. So I think in terms of evasion of innate immunity, coronaviruses in general can evade innate immunity to some extent, definitely. But the three severe ones, meaning MERS, SARS, and SARS-CoV-2, seem to be much more potent.
All right. Another question from Christina. Would you like to unmute yourself and ask your question?
Yeah, sure. Hi, Shiv. I was wondering what's more important in determining the efficacy of vaccines, either T cell or antibody response. And I'm asking this because various vaccine trials have reported different levels of neutralizing antibody levels as opposed to T cells. And I think the AstraZeneca one had more T cell response.
I think if the reality is this, none of the vaccines that have worked in history have worked without making good antibodies. There is no vaccine that is really effective that only creates T cell immunity, right? But to get good antibody immunity, you need T cell help. So there is actually collaboration with T cells and B cells is essential to get a good antibody response. Whether it is even theoretically possible. See, I have close colleagues who are working on T cell vaccines, even for HIV. Whether those CD8 cells clear virally infected cells. So after you got infected, if you want to clear a virus, a CD8 vaccine could be helpful. But if you already got infected, and if you're susceptible and you start to take off, and then you wait for your CD8 cells to be reactivated, you may or may not be protected well. It could work, but it's still going to get infection. It's not like you can't then disseminate a lot of virus and get pretty sick. So it could help someone to have CD8 immunity, T cell immunity. Just to protect yourself, you need to have antibodies that last for a long time. And those you are only going to get from TB collaboration. So there's nothing about, so the way the reports come out, they don't try to tell you about T cells and B cells. There's even a report which came out that looks at TFH cells in the blood. But TFH cells in the blood are the equivalent of what I didn't talk to you about, but I did talk to Alice and Suparna and a few other people who came to class today. is that there are pre-germinal center T follicular helper cells. Pre-germinal center T follicular helper cells are abundant. Okay, they are not a problem in COVID-19. They just can't become true germinal center TFH cells with BCL6. So seeing them in the blood doesn't convince me that you're going to be protected. I think the only correlate that you can get from the blood that will convince me is that if you take plasma blasts 2 weeks after a boost and look for somatic hypermutation. If you get somatic hypermutation, I will then think, okay, likely, since you have a germinal center response, you're probably going to make long-lived plasma cells, which will go to the bone marrow. That's my hope. We don't have any other good correlate.
All right, thank you. And I think we only have time for one more question before we get
I'm
to.
happy. Yeah, okay. My rhyme will take 3 or 4 minutes, and you're free to leave. Okay.
Okay.
So
Okay.
I'm
Great.
not. I'm willing to stay a few more minutes and take your questions. That's more important than the rhyme. Yeah.
Thank you so much. But we would love your rhyme. So I think another thought here is, since you said that lymphopenia is obviously one of the major drivers that we see in patients with COVID-19, do you think that maybe transfusing patients with lymphocytic cells in the future would also be a potential therapy for these patients? What do you think about that?
No, I mean, there's obviously problems about compatibility, right? I mean, that's like doing a transplant rejection. You might create a strong immune response, which will be damaging by just putting, unless you have an identical twin to do it from. So that's not likely to be practical or usable in a large number of people.
Okay. And do you think allogeneic and, differentiating them in vitro and expanding them is also not likely possible, you think?
I mean, if you said, okay, I'll take some of your lymphocytes, I'll differentiate, and before you get very sick, I'll have enough to give them back to you, maybe. I think there are better ways to deal with it than go there as a therapeutic
Right.
angle. I think like using monoclonal antibody mixtures and so on, those things might work earlier on. I think nothing is going to beat good antivirals when they emerge, which target the enzymes that allow RNA replication and so on. There's so many targets in these viruses. But
Yeah, absolutely. Okay, Icon, do you want to ask your question?
Sure. I actually have 3 questions, 2 pertaining to today's talk. Great talk, Shiv. My first question pertains to your conjecture on why there's a lower number of plasmacytoid dendritic cells in the patients who suffer severe clinical course. And the second question is, why are people not investigating the durability of humoral responses in the mRNA vaccine studies?
so, I mean, to really investigate durability, you have to look for a long time. Obviously, the durability will now get investigated after you vaccinate people. The companies want to sell the vaccine, you'll vaccinate people. Then you look after 6 months, 9 months, 1 year, see what the antibody titers are. Then if you're dropping, you'll tell people, no, you need to get vaccinated again. And that's probably good for the company. They can have you vaccinate every year. Okay, but ideally we'd want to get a vaccine that lasts for some time. So I don't know, because there is no data on mRNA vaccines in humans. Okay, that's why I don't know. And the other question was about PDCs. I think there's severe bone marrow suppression of lymphoid-like progenitors differentiating into true lymphoid type cells. And this probably affects right down to the ILCs and PDCs. That's what I think is happening. Whether that's because of some particular cytokine that gets into the blood. This is seen, by the way, all of this is not unique to SARS-CoV-2, by the way, or COVID-19. Any severe viral infection, very severe, like I've told people, if you had Ebola, if you had Marburg, if you had H1N1, similar things happened. And you lost germinal centers, as people didn't document it in any detail. Okay, so I think that's a general phenomenon. And in mice, we can recreate all these phenomena by creating severe infections. So I do think that, the phenomena are real, and the PDC loss is there. Why it happens, it's not good for us. How can we prevent it? If we knew, that could be a therapeutic as well, that prevent bone marrow suppression early, and maybe you'll be protected. That's possible.
My last question is, since there's a growing number of people, I think starting from Hong Kong now into America, there's increasing number of people who are reinfected with the virus. I was curious if it's due to the decreasing number of antibodies or because of that aspartic acid to glycine mutation or a combination of both.
So the mutation you're talking about, the D614G, allows the virus. So basically, when you have these RBDs standing up, the standing up of the RBDs is happening more frequently when you have the D614G mutation, right? So if you stand up an RBD more frequently, if I'm expecting Aikwan knows because he's in the Baruch lab, then it's going to bind to ACE2 more frequently. Okay, so that might be one way it happens, that you actually allow that to happen more. That virus is not necessarily more pathogenic. Okay, and for it to get in, the reason why it's getting, it's not because it's changed the spike structure or the RBD structure. It's getting into people because they don't have antibodies.
All right. And I think we only have one last question here. Raphael, do you want to ask your question?
Yes. Good to see many of you back. I left Harvard some time ago. So my questions were, the two questions. One is about the lymphopenia. If actually it's known their cells are just disappearing, like dying, or they're going to the lungs and to other organs like as infiltrates. And the second one about the germinal center inability to be maintained and to develop is if in your work you looked into T follicular helper and T follicular regulatory cells ratio, like a Peter Sage paper.
Yeah, so I can tell you it's in our paper, and I have data from the mouse as well, which is quite convincing. We have no T follicular regulatory cells in patients. They have no germinal centers. We can't find T follicular regulatory cells. In the mouse, we are able to now look early on, and even before germinal centers are formed, first of all, TFH cells are crippled and TFRs are also gone. It's quite dramatic because in the mouse with FOXP3 and looking with CXCR5 and everything, you can look at TFRs quite well. I didn't realize it would be as easy as it's turned out. And they are gone very early. So I don't think the loss of TFRs is the driver. So Alicia has a question. Could reinfection be actually, say that again, Alicia?
This has nothing to do with like literature that I read, but like I don't know, like in Italy currently, many people are tested positive again after a few months of being tested positive for the first time, and many of these reinfection are being labeled as like a reactivation of a residual primary infection, and I was wondering if that was possible.
Well, so the ones that are being called reinfection today are when you could clone the virus in each case, sequence the whole virus, and show it's a completely different virus which would not meet the natural mutation rate that would occur had it remained in you. So only then are people claiming reinfection. If you just do a test and find the virus again, it could be dead virus that's persisted. But if you find full-length virus and it is not the same virus because it's different at so many changes that you see, only then are people calling reinfection. So we are underestimating reinfections today because it's hard to find them. You have to do all of this stuff. Doesn't mean everybody's getting reinfected. Okay, I don't want to give you that impression. It doesn't mean, by the way, you are probably protected for some months after infection, okay? Because you get a very mild disease, mild infection, you don't get much antibodies. But if you've got relatively either moderate or severe disease and you survived it, you're probably protected for some time. The question is how long, not yet tested, but you don't have germinal centers. So I think it's going to be like SARS and MERS. If you, during convalescence, you actually, you recovered and you went through convalescence, you might recover your germinal centers and then you might make some good long-lived antibodies, but that will be in a small number of people.
All right. I think that was a wonderful discussion. We're ready for your grand.
Okay, I'll just do this. For those who have heard this before, I have changed one line. It's not a lot, but that's it. I call it quietly into the night. My noble cousin expresses CD4. He is a veritable cytokine store. But a DC's come hither and he's ready to go. His CD40 ligand he just loves to show. My cousin's promiscuous, hits on passers-by, kisses the wrong B cells, then tells them to die. The right BCR can give fast the slip, shuts off caspase 8 by turning on flip. I grew up a cell of the CD8 kind. I was naive and aimless, bored out of my mind. I wandered like a cloud from node to node. CCL21 determined my abode. Along came a virus, infected someone young. SARS-CoV-2 can damage the lung. My DC friends each picked up a sliver, and in the lymph node, I started to quiver. Is this the feeling that Ben had for Jen? Or when Romeo saw Juliet again and again? Peptide and class 1, and then some B7. I leap in delight. I'm in co-stimulatory heaven. I'm no longer a boring CD8 cell. I'm a killer now, baby. A real CTL. No fleeting feelings like Jen had for Ben. I proliferate like crazy again and again. The liver beckons to me, and my brothers as well, I meant to say lung. We each form a synapse with an infected cell. With perforins and granzymes, the little clefts we fill. Oh, what passion to lock lips, but kill. Passion we have now, but once we did grieve. For siblings lost, forced to take their leave. They perished at the double positive stage. Cell structures they did strongly engage. They made some BIM and then felt weak. Their mitochondria started to leak. Cytochrome C bound to APAF1. Caspase 9 began to hum. We wept, but they chose not to fight. They vanished quietly into the night. COVID-19 is a tragic disease. I don't want to give you any other impression, but on that tragic note, I'm going to leave all of you. Okay?
Wow. Thank you so much, Dr. Pillai. I'm sure everyone enjoyed that. Thank you for your time.
Most welcome. Bye.
Bye. Thank you so much.
Bye.
Everyone, so we just want to remind you guys for our next talk in November with Dilip Moni, and I think it's going to be about adapting education and research for the pandemic. Thank you for attending, and hope you guys have a great rest of your day.